Hello, this is Dr. Mobeen. So we are working on immunology, and today we are working on the question number 10. Question number 10, the way I have done it, and it's just a try to see which one, how the questions should be presented. So I have done it in two ways. One is a longer version where I've explained everything concerning this question, and then there is a shorter version, which is this version, where I'm trying to just give a brief commentary for what is the question and what are the answers and what is the right answer. So let's very quickly look at the question itself. The question says, 29-year-old nurse is exposed to a patient with active tuberculosis. So a medical staff, a medical professional has become exposed to someone who had active tuberculosis. She presents to you with a chest x-ray showing a small calcification. So some sort of a healed infection, signs of that. She is otherwise asymptomatic, which also is an indication that she is probably not having an active disease, although we have to do more tests to figure it out, but at least from the question, that is what we are led to believe. Which of the following cells combination is the major contributor to contain this infection? So the key word here is the contain this infection. So which cells here, the ones which are given in the option, which pair of cells, which two cells, which partner cells work together to contain the Mycobacterium tuberculosis? That is the primary question. So now let's see what are the options here. The option A is dendritic cell and B lymphocytes. Option B is natural killer cell with CD8 T lymphocytes. I hope you know that the CD8 T lymphocytes are cytotoxic T lymphocytes. CD4 T lymphocytes with macrophages. CD4, I hope again that you know that CD4 means helper T cells. CD4 T lymphocytes with B lymphocytes. So again, helper T lymphocytes and B lymphocyte. So let's very quickly try to work with this question and see what are the answers. So again, in the other video I have explained this thing in detail. Here very quickly, let's see the option number A, dendritic cell and B lymphocytes. Dendritic cells are very important. They are antigen presenting cells, but in case of tuberculosis, dendritic cells antigen presentation is less important, number one. Number two, why? What is more important? Macrophages are actually more important as APCs for tuberculosis. Second thing is this, that dendritic cells presenting the, so if we have some dendritic cells here which are going to be, let's say this is a cell, actually I make dendritic cells like this. So let's say this is a dendritic cell. If this cell is presenting antigens to B cell, then in turn the B cell is going to release what? It is going to release antibodies. This is humoral response, a chemical response to a pathogen, and Mycobacterium tuberculosis is actually not contained via this chemical response. So dendritic cell can have a role in containing tuberculosis by being a presentation cell, but really in case of TB, macrophage is a more important presentation cell, and then B cells actually have very little to do with the containment of tuberculosis. Humoral response actually does not contain tuberculosis. Actually, if somebody asked you this question, that a person who got exposed to the Mycobacterium tuberculosis or Mycobacterium leprae and then developed lepromatous leprosy, what was wrong? And your answer should be that the person's immune system, so that is a genetic anomaly, the person's immune system, instead of going towards a cellular response, has gone towards a humoral response, and humoral response does not contain the Mycobacterium tuberculosis very successfully. So this actually is a case of healed infection, and because it is a case of actually not active or contained infection, if not healed, then this option is not plausible. So A is gone from our choices. B, natural killer cells with cytotoxic lymphocytes. Now natural killer cell and cytotoxic lymphocytes actually have nothing to do with each other. That is not a pair which works like a pair. Natural killer cells are actually a type of lymphocytes which break rank from the lymphocytes and become part of the innate immune system and are usually taking care of the cells infected with viruses and cancers. On the other hand, cytotoxic T lymphocytes do have some sort of an involvement in tuberculosis here, because if you see, when macrophage takes Mycobacterium tuberculosis, phagocytosis is, and then there is of course phagolysosome is formed and the bacteria is broken up and antigen is presented. As part of the APC function in the macrophage also releases interleukin 12. That interleukin 12 activates T helper 0 or causes T helper 0 to become T helper 1. T helper 1, this CD4 cell, then in turn releases IL-2, which causes cytotoxic T cells to become active. So you could say that cytotoxic T cell could have something to do over here, but honestly the cytotoxic T cell, other than killing some of the macrophages which contain the pathogen in them, there is no other function of the cytotoxic T lymphocyte for containing the Mycobacterium tuberculosis. The major cell which does this function is this pathway, the major pathway which works here in the cells. The T helper 1 gamma interferon acts on the macrophage, activates the macrophage, and what does the activation of a macrophage mean? Receptor exposure is reduced, or receptor expression is reduced. Oxygen species, nitrogen species are increased, reactive oxygen and nitrogen species are increased, that increases killing, and secretions from the macrophage increase, which causes the chemotaxis and other such functions. Even for the granuloma formation, the macrophage secretions are necessary after this activation. So the option number 3, CD4 T lymphocytes with macrophages, can these be the ones? Yes, this is a CD4 T lymphocyte. So if you come here, this guy is a CD4 T lymphocyte, and he is working, or this is working with a macrophage. So yes, macrophage and CD4 T lymphocyte, this process, so these two guys are partner cells. So C is definitely a plausible answer. Let's see what is in D. D says helper cell with B lymphocyte. So remember, helper cell with B lymphocyte is the humoral pathway. So what happens is, if we have IL-4 coming to the naive helper cell, then the helper cell would become T helper 2, and that T helper 2 would then release IL-4 and IL-5 and work on the B cells to start producing the humoral response. So that response, of course, in case of Mycobacterium tuberculosis, is not the right response. So the option number 4, CD4 T lymphocytes with B lymphocytes, is actually not a correct option. So in this particular question then, if we go back to the question, in this particular question, the option which is important is the C option, or the option which is correct is the C option. So we would then from here go. So again, this is a case where macrophages and the T helper cell 1 work together to contain this infection. They form granulomas, they kill the bacteria, and they go from there. You should also know how a granuloma is formed. Very briefly, we have in the functioning of the granuloma, when the secretions come from the macrophage and other cells and the cell breakdown products, those secretions cause chemotaxis, neutrophils come in, monocytes come in. Within 2 to 3 days, you would see those cells coming in and present in that area. Within 5 to 7 days, you would see that there would be macrophages, mature macrophages, and then epithelioid cells will be forming. And then after 9 days or so, you would see that there will be helper T cells, there will be fibroblasts present, there would be the collagen or extracellular matrix has been secreted there as well. Because of gelatinase and collagenase, there is caseating necrosis started in the center. The tumor necrosis factor alpha has been released, that also causes the caseating necrosis in the center of the granuloma. So that all is really the granuloma formation, and when the cells break down in there, the calcium pours in, and that is what would end up in the calcification. So this is macrophage and CD4 helper cell interacting together to contain Mycobacterium tuberculosis. Thank you very much.